So welcome to the CADASIL Connection webinar series. I'm Bert Kosiski, a physician at Hennepin County Medical Center and a professor of medicine at the University of Minnesota in Minneapolis, Minnesota. I am moderating this session in support of cureCADASIL, not because I'm an expert in CADASIL, but because I have a family member with CADASIL. Today's webinar is produced in conjunction with the American Society for Neurotherapy and Repair. Trying to get the slides to change here. There. cureCADASIL is a nonprofit organization dedicated to helping patients, families, caregivers, and other supporters touched by CADASIL. We aim to unite patients and the medical community toward the common goal of treatments and ultimately a cure for this rare genetic disease by promoting communication, advocacy, research, and education. The president of cureCADASIL is Dr. Jane Gunther, and many thanks to her for helping to mentor me in setting up this webinar and in everything. So here to say a word about the American Society for Neurotherapy and Repair is their current president, Dr. Liru Zhao, who is professor of neurosurgery at State University of New York, Upstate Medical University, Syracuse, New York. Dr. Zhao?
I'm sorry, I cannot see your slides.
I'm on the show screen. Let me click it again. And
yeah, okay. Thank you, Bert, for your introduction. Now I would like to take this opportunity to introduce ASNTR. The American Society for Neurotherapy and Repair is a non-profit organization devoted to scientific and educational purposes. The ASNTR is dedicated to the development of pioneer reparative therapies to treat neurological disorders, neurodegenerative diseases, and injuries in the brain and the spinal cord. This goal is achieved through utilization of novel cellular therapy, gene therapy, molecular therapy, and other biological therapies. ASNTR members include scientific visionaries and developing scientists who work together in a collegial and collaborative manner. If you want to learn more about ASNTR, please go to our website www.asntr.org. Next slide, please.
is the slide changing for you?
No, sorry.
Let me go back and forth. How about now?
Oh yeah, okay. Now I also want to share the information with you about the 28th annual conference of ASNTR, which will be held August 19 to 22 this year in Clearwater Beach, Florida, in the United States. In this conference, we have a special section for CADASIL, exchanging research updates in pathogenesis and the therapeutic strategies for CADASIL-caused stroke and vascular dementia. We also have a special plan to host a session of Alzheimer's disease-caused dementia in the same day with the CADASIL session. The purpose of having these two sessions in the same day is to explore there's any pathophysiological similarity between these two types of dementia, and to discuss any potential therapeutic target and therapeutic strategy for these two types of dementia. You're all welcome to attend the ASNTR 2021 meeting. The general abstract submission deadline will be announced soon. We have an education and training program to support about 20 to 25 outstanding students and the trainees to attend this meeting by travel world. Students and the trainees include undergraduate students, graduate students, postdocs, clinical research fellows, and residents. Both national and international students and trainees can apply for this travel award. The deadline for submit travel award abstract and application will also be announced soon. Please check update information through our website. Next slide, please. Okay, I would like also give a brief introduction about my lab research. The research of my lab is focused on exploring pathogenesis or pathological progression and brain repair strategy and mechanism for stroke, CADASIL, Alzheimer's disease, and traumatic brain injury. Blood clots, blood vessel, and blood flow, which has a medical term, is called thrombosis. A recent study in CADASIL demonstrates that capillary thrombosis occurs in the brain of CADASIL. We also observe that cerebral capillary thrombosis caused ischemic neural loss is attenuated by repeated treatments of stem cell factor and granular cell stimulating granular cell colony stimulating factor in transgenic mouse model of CADASIL. Here are representative 3D confocal images showing capillary thrombosis in the cortex. The capillary wall is visualized by the red color, which is CD31 positive endothelial cells. The green color is green fluorescent protein labeled blood cells. As we can see, GFP labeled blood cells completely blocked the capillaries in the cortex. So when blood clot completely blocked the capillaries, it will lead to ischemic damage to the local neurons. So we then quantified the neurons surrounding the thrombotic capillaries in the cortex of NAGLU3 mutated mice treated with or without SF plus SF. Our data shows that the neurons next to the thrombotic capillaries significantly increased by SF plus SF treatment. The pathogenic mechanism of cerebral capillary thrombosis and the reparative mechanism of SF plus SF treatment are not clear, that are being investigated in my lab. I finish up here, and thank you so much for your attention.
Thank you very much, Dr. Zhao. Internet's slow today. I don't know why I can't seem to change slide. There. So, sorry for the technical difficulties, but it gives me great pleasure to introduce today's speaker. Fabrice Dabertrand is recipient of the 2019 $1 million bike ride grant made possible by University of Pennsylvania, cureCADASIL, and the generous donations of the CADASIL community. The $1 million bike ride supports one-year grants for rare disease research. With these seed grants, researchers can produce supporting data that enable them to be competitive for larger grants to continue their much needed research on CADASIL. So Dr. Dabertrand received his PhD degree in 2006 in neuroscience and neuropharmacology at the University of Bordeaux in Ségolène, France. And in 2009, he did a postdoctoral fellowship in pharmacology at the Lerner College of Medicine at the University of Vermont. In 2015, he received the New Investigator Award of the American Physiological Society, a very prestigious award. In 2017, he received an NIH grant as a principal investigator to study capillary control of cerebral blood flow and its disruption in small vessel disease. And then he moved to Colorado, where in 2018 he became an associate professor in anesthesiology and pharmacology departments at the University of Colorado. So today's topic is restoring cerebral blood flow in a CADASIL model. And years before the emergence of strokes or mental decline, patients with CADASIL show deterioration in the brain's ability to increase blood flow in response to appropriate signals from the nervous system. Using a genetic mouse model of CADASIL, Dr. Dabertrand and co-workers determined the abnormality to be located in cells lining the small blood vessels or capillaries. They found that CADASIL is associated with reduced production of the phospholipid phosphoinositol 4,5-bisphosphonate, or PIP for short. This prevents the cells lining capillaries from sensing brain activity and causing small arteries upstream to relax and increase blood flow locally. They found that injection into the bloodstream of PIP2 corrects this problem in CADASIL mice and restores blood flow to the brain in response to neuron activation. I'm going to turn the screen over to Dr. Dabertrand. I do want to say that you will have opportunities to ask questions. And if along the way in the presentation, if you go to the question panel that you'll see on the right side of your screen, you can type in questions and we'll try to get to them at the end of his talk. And so,
thank you so much, Bert, for the introduction. And good morning, everyone, or good afternoon for our friends in the East Coast and country. Cheers for all the people in Europe. It's already happy hour there. And I'm really happy to be here with you. And I really want to thank you and the committee for allowing me to present this data. This kind of talk with the patients, the family, and the professional is always a bit challenging because we don't want to knock anybody out with a hard dry science, but in the same time we need to deliver a complicated message. So I packed my talk with a bunch of movies, so feel free to relax, and if my French accent is too annoying, just turn my voice off, put some music on, and enjoy the movies. Okay, let's rock and roll. Today we're going to talk a lot about neurovascular coupling and functional hyperemia. And I'd like to try start with a bit of definition and vascular historical perspective. Neurovascular coupling refers to the ability of the brain to locally adapt the blood flow delivery to the neuronal activity, because the brain has little energetic reserve. It's an on-demand system, and it is illustrated here in a historical experiment performed by the Italian physician and physiologist Angelo Mosso at the end of the 19th century. His patient, Michele Bertino, following a traumatic injury, was left with a thin layer of skin covering a gap in his skull. And using 2 sigmographs, Angelo Mosso was able to simultaneously measure the variation of blood volume in Michele's arm, here, this is the lower trace, and the brain over the window here, which is the upper trace. And you can see like this little variation are actually the heartbeats. So it was very, very precise instrument at the time. And as you can see here, when Angelo Mosso asked Michele Bertino to calculate 8 times 12, the blood volume in the brain increased, but not so much in the arm, as Michele produced a mental effort to calculate that. And a few years later, Roy and Sherrington wrote in an historical paper a statement that remains true today, that the brain possesses intrinsic mechanism by which its vascular supply can be varied locally. This is a key word here, in correspondence with local variations of functional activity. And these properties are today's basis for the functional MRI that we use in this machine that allow us to visualize the brain activity. I'm sure you've already seen some kind of these images, and here is the effect of number size calculation by abacus expert. You can see that when they do an operation with 1 digit, they have a little neuroactivity, which is actually the oxygen consumption by the neurons, and which corresponds to the increase in blood flow. And as we get more complicated operation, like with 3 digits, the brain consumes more energy, and 6 digits even more. And the key question is, how is neuroactivity translated into increased local blood flow, and how cerebral vascular disease impair neurovascular coupling? And to answer this question, we need to first understand the topology of the brain vasculature, because it creates really distinct zones. And first, it should be noted that this organization is kind of similar between the different species. Here is the cartoon from the human brain, the cortex. Here's from the rhesus monkey, and here is from the mouse. So if you look at the vasculature of the brain, it's not like the mouse brain is a mini human brain, but more like a chunk of it, because the law of diffusions are essentially the same. So, and the red blood cells at the end of the day are very, very, very similar in diameter. So what we have is on the surface, we have the pill vessel, arteries and veins that are very rich in anastomosis. And the arteries dive into the cortex with one right angle here, and without any collateral supply, meaning that each of these arteries perfuse a discrete volume of brain, and this creates a bottleneck effect that makes each column of the brain parenchyma served by a given arteriole. But that also makes this region a really important regulator of blood flow, but also a region especially vulnerable to insult during cerebrovascular disease. So now we know where the blood flow can be regulated, can understand how. This is an isolated and pressurized with this cannula inside here. Mouse intracerebral arteriole. This is very tiny, about 20 micron diameter. This is about a third of a human hair. And part of my postdoctoral training in Mark Nelson lab in Vermont was to adapt imaging system to mouse cerebral circulation. And as we can see in this movie accelerated 10 times, when you take the arteriole out of the brain and pressurize it to mimic the blood pressure, the arteriole constricts in response to this pressure. It's a feminine term, myogenic tone. It was described by Baylis at the beginning of the 20th century. It's a very important mechanism, and the consensus in the field is that the blood pressure creates a graded membrane potential of the smooth muscle, a graded depolarization, sorry, of the membrane potential of the smooth muscle, and that open voltage-gated calcium channel, and then the calcium goes into the smooth muscle cells and create this constriction. Myogenic tone is an intrinsic and fundamental feature of small arteries and arteriole, and that it protects the capillary for disruptive high blood pressure. And we regularly experience the importance of myogenic tone when we, for example, stand up too quickly and our vascular system was kind of quiescent, and blood tanks in the guts and the legs, and it creates a drop in pressure in the brain, and we feel dizzy. And as the arteriole constrict and bring back the pressure in the cerebral circulation, we feel better. It must be noted that in the cerebral microcirculation, it provides also the vasodilatry reserve that is necessary for an effective neurovascular coupling. And you don't have to understand everything here. Just what I want to highlight is the resistance created by the flow, by the arterioles, the diameter, is inversely proportional to the force power of the radius of the arteriole. Meaning that if you have a small dilation, you actually have a huge drop in the resistance and then create an increase in blood flow locally. And one last thing I want to highlight here is the myogenic tone. When we do it like this ex vivo, the prep has to be perfect. So seeing the arteriole developing a myogenic response is for us a very good indicator of the quality of our experiment, because if you don't do things right, you never get this kind of pre-required. Another important aspect is the endothelial cells. So they line up the lumen of the arteriole, and they provide an hyperpolarizing influence that oppose the membrane depolarization and so create a vasodilation. Here's in real time the effect of a VAS application of a potassium channel agonist, especially for the SK and SK channel, the NS309. And when the drug hit the VAS, you can see in real time how fast the arteriole dilate. What is interesting is the endothelial cells not only line the lumen of the arteriole, but they also further down the capillary, the vascular tree, they form the capillaries, which have been thought for a very, very long time to be the site of exchange of glucose, oxygen, pick up the CO2. So we have this brain vascular wiring that you can appreciate here, where all the nervous tissue have been removed, and while the arteriole create the bottleneck for blood delivery, the capillary vastly extend the perfusion territory, and it's estimated that in our brain we have about 100 miles of capillary. So we're really talking about billions of cells, something like neurons. It's not something we really are always aware of. And like I said, for a long time, capillary have been thought to be passive tube, but this architecture really invite the hypothesis that the capillary creates a sensory web that could be able to detect and initiate vasodilatory signal that can travel upstream to the arteriole to regulate the blood supply. And these anatomical features really show the importance of the vasculature. And it's shown even more during pathological condition, unfortunately, like stroke, which generally is an occlusion of this more arterial here, and dementia. These two pathological conditions are right now one of the most pressing health issues in the United States. Stroke costs about $34 billion, accounting for 1/5 of deaths, and is a leading cause of serious long-term disability. Vascular dementia as well, people not necessarily realize, but in 2012, the World Health Organization estimated that 35.6 million of people will live with dementia, a number that is anticipated to triple by 2050. And it makes the vascular dementia the second leading cause of dementia, about 30%. And even if you take the leading cause, which is Alzheimer's disease, the vascular component of it is thought to be more and more to be a very, very important adjuvant of the disease progression and the neurodegenerative process. So, but despite this profound impact on human health, we don't really have treatment at the time. And in a clinical perspective, people have grouped stroke and vascular dementia under a small vessel disease of the brain umbrella that are characterized by a group of common symptoms, especially progressively many years before becoming clinically symptomatic, because the brain has this huge plasticity and ability to adapt until it reaches a condition that it can compensate anymore. And it's characterized by enlarged perivascular space, white matter lesion on the brain MRI. And among this group of diseases, one, as you know, is particularly frequent, is CADASIL. It's the most frequent hereditary form of SVT. And it's an autosomal dominant disease, which is caused by mutation in the NOTCH3 extracellular domain, which accumulate around the vessels, and it targets smooth muscle cells, pericytes, because these two cell types express NOTCH3 receptors, but also, like I'm going to show you, even endothelial cells in the capillaries. And of course, it's an enormous burden that we are going to study, but it's also very important for us scientists to examine this disease, because as stated by Niccolo Machiavelli here, it's actually a quote he used to describe corruption in society. But he used a medical metaphor. What physicians say about disease is applicable here. At the beginning of the disease is easy to cure but difficult to diagnose. But as time passes, not having been treated or recognized at the outset, it becomes easy to diagnose but difficult to cure. And with CADASIL, because it's caused by mutation, we know that in people before the cognitive deficit that lead to dementia and even the ischemic stroke, we know that even before this devastating episodes, we observe a decrease in cerebral reactivity. And it's been studied published recently by Hugues Chabrier group, probably one of the world experts on CADASIL, where they look at people with CADASIL and look at neurovascular coupling and functional hyperemia. In people, I think the average age was about 43 years old. And so it was really before any cognitive decline, before any stroke. And they subjected the patient to flickering task and hand task, and look at the blood flow increase in region during this task. And very, very interestingly, the visual stimulation in both cases, in short and longer term stimulation, they reported a decrease in the ability of the brain to increase the cerebral blood flow, where the neuron were activated. And same thing with the motor task, although it was not significant with the shorter stimulation, but still with the longer stimulation. So it suggests that the impairment of the neurovascular coupling, the decrease in the ability of the brain to increase blood flow in response to neural activity is a very, very early event in the disease progression. And it's something that we already knew from our mouse model. Mouse model have been developed by Andrew Tell, another world expert on the disease. And we observed that actually at around 6 months, which of age, these mice, which will be about 37 years old in people, show reduced myogenic tone in cerebral area and also an attenuated functional hyperemia in young CADASIL mouse. And we have studied this impairment extensively for the last 10 years. And this is one of the emerging concepts is that in physiological condition, on the surface of the cells, you have a metalloprotease called ADAM17 that cleave the pro-HBEGF. This is a pre-ligand of the EGF receptor, and this is an ongoing mechanism called shedding that our cells continuously do. They produce this pro-ligand, it's cleave, and it activates the receptor, and it keeps our cells happy, communicate with each other, and metabolism high. What we think happened in CADASIL is the NOTCH3 accumulate around the vessels, and you recruit and accumulate more and more TIM3. And the TIM3 is a specific inhibitor of the ADAM17, and so you shut down this signaling pathway. And we observe that actually when we introduce HBEGF externally, we are able to restore myogenic tone and neurovascular coupling. But we were missing some key element to understand this effect on vascular coupling. And it's only recently that we made this hypothesis that the brain capillary, as I said, may act as a sensory web to detect and respond to neuroactivity through electrical and calcium signaling, picking up, for example, I'm going to talk about potassium ions increase and transducing that into a vasodilatatory signal that travels upstream and goes up to the arteriole to cause a dilation and increase local blood flow. So why potassium here? Well, it's well known for the people in neuroscience that when neurons fire action potential, membrane conductance for sodium increase a lot with an inward current. So basically sodium ion going into the axons of the neurons, and the repolarizing phase include potassic outward conductance, meaning that the potassium will go out of the axons and accumulate in the extra cellular space. And here's, for example, a recording from the 90s showing the potassium concentration going from 3 millimolar to up to 10. It's also, potassium can be also a good neurovascular coupling agent because it's really rapid on-off kinetics, and it's known to activate a channel that I'm going to talk a lot today, which is the strong inward rectifier potassium channel, or KR 2.1. Here's an example of a potassium in action on arterioles. Again, this is an arteriole isolated from mouse brain. And here is the recording of the diameter of this arteriole. So when it increases, it means it dilates, and when it decreases, it means it's constricted. Raising external potassium from 3 to 8, 15, 20 millimeter of potassium causes almost maximal dilation by stimulating the KR channel. These channels are tetrameric channels. Here's the alpha subunit, and they are known to be present in the smooth muscle and the endocellular of parenchymal arteriole. And as raising external potassium fusion brings the potassium equilibrium, EK, closer to the resting membrane potential, here you can see the arteriole reconstrict, and with 60 millimolar, even cause a depolarization that induces the constriction. And KR channel have this unique property of being activated by both external potassium, but also membrane hyperpolarization itself. So these two facets of KR behavior synergize in response to a small increase in external potassium, leading to a dramatic feed-forward action, bringing plasma membrane to EK and create this dilation. And so you can see that KR is some kind of potassium electrode in the brain vasculature sensing external potassium and hyperpolarizing cells in this response. And so we developed this model that actually capillary endothelial cells may express KR channel, sense the increase in potassium due to neural activity and action potential, and hyperpolarize. And through gap junction, this hyperpolarization can be transmitted to the adjacent cell, and in a regenerative mechanism, travel upstream up to the arteriole and cause the smooth muscle cells hyperpolarization, decrease the open probability of the voltage-gated calcium channel, and cause the relaxation. And I'm going to show you some data that was supporting this mechanism. It was published a couple of years ago with my great friend Tom Longden from the Nelson Lab, and well, actually on the cover in Nature Neuroscience. And to study this mechanism, part I did in this study was to being able to isolate again the vascular from the mouse brain with here what is an arteriole visible in red due to the genetic expression of actin and cherry in smooth muscle cells, and still attach about 300 micrometer long capillary here stained with lactate. And our key experiment was to pressurize this vessel, seal everything, so we have a pressurized system, and locally apply potassium here while tracking the diameter there. And that's exactly how it looks like. Here's a pipette filled with 10 millimolar of potassium. It is completely ex vivo. There is upstream, there's a cannulation procedure to create blood pressure, and here are the capillaries. You can see some red blood cells lying in the bottom of the chamber. So it's good for scale. You can say that this diameter here is about 15 micron. And what happened when we lower the pipette and stimulate here the capillaries? Well, the arteriole dilates. This is accelerated ×2, but you can appreciate how fast it is. The onset of the response is about 3 seconds after the beginning of the stimulation, and the plateau is reached before 6 seconds. It's sustainable, it's repeatable, it's a very challenging experiment, but when it works extremely well. Now, what is interesting is we did the same thing in our CADASIL mouse model, and we thought that nothing was happening. So something was not communicating the hyperpolarization, because when we stimulate the arteriole itself and the KR there was still an ability of the arteriole to respond, but the signaling between the capillary to the arteriole was shut down. So we were really interested in this observation, and to further investigate it actually in vivo, we've used this technology called multiphoton fluorescence microscopy. This allows us to do intravital recordings of the blood flow in the brain, because if you stimulate a fluorophore at a certain wavelength, there's a little bit of penetration within the tissue, but not so much. But actually, when you send 2 photons at wavelengths that's half what you normally would have to excite a fluorophore, it can deliver the same level of energy, but with a much better tissue penetration. And it also creates, interestingly, a confocal imaging, means like you really are imaging for one focal plane and not the entire volume. So we do a cranial window on the mouse, and we inject in the vasculature dextran with FITC or TRITC, something that glows basically, to visualize the vasculature. And here is the map with showing the surface here of the brain with vein and arterioles at 0 micrometer. And the red is actually showing how deep you can go, up to 350 micrometers. It's an image made by my good friend Tom Longden. And when you image, you get this kind of resolution through the cranial window going down. And what you see in black moving here are actually the red blood cells, because the capillary, they don't take the dye, the FITC dextran, so they appear black, exactly like a car passing on top of a glowing road, for example. And since the capillary size is about less than 5 micron and the red blood cells about 6 micron, only one red blood cell can go through the capillary at a time. And what we can do, we can introduce a pipette, again, filled with 10 millimolar potassium, and deliver locally the potassium next to the arteriole, and see what happens. So to do that, we basically count red blood cells going through the capillary. We do it at high speed with a line scanning technique. So each red blood cells going through is one of these stripes, and the higher the angle is, meaning the speed of the red blood cells is going faster. And if you perf the potassium around the capillary, you can get a sense of a upstream dilation by looking at the red blood cells flux that is actually increasing in response to stimulation. And again, when we did this experiment in the CADASIL mouse, the increase that we saw in the control was just not there. So it looks like confirmed that there was no communication between the capillaries, the stimulation with potassium, and the upstream dilation. Masayo Cody, another really talented scientist in Vermont, this made this really interesting experiment where she stimulated the whisker of the mouse and record the increase in cerebral blood flow response. So in control, we observe this local increase in blood flow from functional hyperemia. This is highly sensitive to barium. A big component, big part of it is inhibited by barium. Why it is? Because barium actually is a pore blocker of the KR here. And indeed, in CADASIL, you still get a little bit of response into whisker stimulation, but the barium has no effect, meaning that this activation of KR is actually missing. So there's no barium sensitive component. Why is that? Why we don't see this activation? Well, there's a good friend and very talented scientist, Osama Haraz in Vermont, look at the current density of isolated cells from the capillary. So it's a very challenging experiment. You basically isolate the cells freshly from the mouse brain, and with a micropipette, you form a seal with the membrane, and you get access to the electrical behavior of the cell this way. And what you recorded with the appropriate condition and voltage range was that the current density attributed to KR channel in these endothelial cells were about half of what we in CADASIL of what we observed in control mice. Very, very interesting observation. But I eventually raise a question: what is a 50% reduction in KR current can do? Is it sufficient to completely shut down, or is it not? And we answer this question with different experiments, but one is I think very, very interesting, and I don't want to detail that too much. But when you look at the conductance of the KR channel between 3K and 10K, and the background current here, you can see you create a switch when you raise 10K, 3K to 10K. You created this second point intersection, and that corresponds to the hyperpolarizing jump of the arteriole in response to potassium challenge. And this is simulated in silico by our collaborator Nitsukias and Arash in Florida International University. But if you decrease by 50% this current, you lose actually this second point intersection, and the whole network then failed to make this hyperpolarizing jump to actually create this regenerative vasodilatorate signal, and the system stays at the membrane potential it was even with the potassium challenge. And so why was the current decrease? And one thing we hypothesized was it was linked to something that is very well known in the field of people studying the KR channel, is his association with the phosphatidyl 4,5 biphosphate, or PIP2. It's a minor phospholipid in the membrane, in the inner leaflet of the membrane, and it needs to be bound to the KR channel to make it activable. Without it, the channel is in conformation that is not activable by potassium or hyperpolarization. And what is remarkable about PIP2, it's in constant balance between breakdown and synthesis in the cell. It's consumed by activation of many different processes, notably the GQ-PCR, and activation of phospholipase C that cleave the PIP2 into the AG and IP3 receptors, well-known and canonical signaling pathway. And it's synthesized by the PI4 kinase and PI5 kinase using ATP, ATP being the energetic source of the cells. And interestingly enough, well, we thought that in CADASIL mouse, the ATP in endothelial cells happen to be likely lower compared to wild type. And this lower metabolic states was actually enhanced when we apply it to each year. Very, very interesting. So what Osama did was reintroducing PIP2 in the bath of the cells when doing patching them. And without PIP2, the cells, as I said, show a rather low current density. But introducing PIP2 in the bath solution, presumably was taken up by the cells and rearranged and associated with KR, showing an increase in current density, meaning that the channel was there. It was not just in the ideal configuration to function and develop current. And so providing an infinite external source of PIP2 was enough to restore this current density. And remarkably, PIP2, when we did the same experiment on control capillary endothelial cells, did nothing, and same thing when applied onto the arterial, it didn't change the magnetic tone, showing that it was really a deficit that was located into the capillary endothelial cells. So we do this experiment again, like I showed you before, stimulating with the capillaries 10K, didn't do anything. Again, internal control, still ability to restore the dilation by stimulating the arterial directly. But then we introduce a soluble form of PIP2 at a rather high concentration, 10 micromolar, and boom, we'll be able to restore the dilation back by stimulating arterial with 10 million potassium. I can show it again. It's really remarkable. In 15 minutes, being able to correct the deficit that's been developing in mouse. Remind you that these are 6 months old mice. Within minutes is truly something remarkable, how fast it can be acting. And here's the summary data, whereas the restoration of dilation induced by PIP2 in response to capillary stimulation. And in the mouse that is genetically deleted especially in the endothelial cells for KR2.1, that basically show the same defect at CADASIL in terms of neurovascular coping. PIP2 had no effect. And finally, Marcelo did this experiment with the whisker stimulation, where she did the whisker stimulation, then inject intravenously PIP2 at 0.5 milligram per kilogram of mouse. And you can see this really significant increase in blood flow that is completely abolished by barium, suggesting that it is indeed the reactivation of the KR in the capillary endothelial cells. So all of these data just got published yesterday in the prestigious Journal of the National Academy of Science, and I'll be very happy to share it with anyone who wanted. Just shoot me an email and I'll send you the study. So I just would like to make this temporary conclusion here that some take-home messages that the brain capillaries act as a sensory web to detect and respond to neural activity, and the cerebral small diseases happen to target these endothelial cells, which turn off electrical signaling during CADASIL. And it's very, very interesting because so far CADASIL has been seen as a mostly smooth muscle cell disease, and while other forms of small vessel disease, the consensus is that the endothelial dysfunction is present, something that's never been reported in CADASIL. So I think here we really put our finger on something very important in terms of disease progression. And we've been very happy to share with you our observation that exogenous addition of cerebral PIP to restore KR channel current density in capillary endothelial cells, and enable normal functional hyperemia. We submitted this observation as a patent, actually. And in the last minutes I would like to emphasize a pet project I really have in the lab right now. Something we call pericyte contractility in CADASIL, and it's some project that I have been in mind for quite some time and never really get the chance to start it up. And when I thought the calls for application from the Milan background, I thought that would be really the perfect way to initiate this project in the lab. And I really want to thank all the people and the cureCADASIL who organize and donate to this grant, because for us in the lab it's been really an exciting project. And unfortunately, it started during last year during the pandemic. So it has starting a seed project, you know, in the pandemic is not exactly the best condition, but we've been able to do so. And one of the rotation students we have in the lab, Daniel Giffrey, really latched on this project and will actually join the lab and keep pushing this project forwards in the next years. So I want to mention that how important it is, your support is not only to be able to make experiments, but it's also helps to foster the next generation scientists. So what are pericytes? Well, it's a very controversial question in the field, but basically pericytes are neural cells that covers the capillaries in the brain. So you can see here the smooth muscle cells in green forming the arteriole, and then when you transition to the capillary, you can see in red this protrudent cells body. We call them, they've been described as bump on the logs, and they do a lot of things. That they are necessary for blood-brain barrier integrity. They also probably shape the network. And recently, in a very, very important paper in Nature in 2019, they showed that actually in Alzheimer's disease, part of the issue is that pericytes constrict and completely shut down the capillary blood flow. And as I said before, pericytes, they express NOTCH3, and they also express TIM3. And however, we don't know much about this effect. We don't know much about what's the effect of CADASIL causing mutation on pericytes, especially pericyte contractility. And one of the reasons why is it's really challenging to study parasites. You know, they are very, very tiny cells, very fragile. And thanks to our progresses in neurovascular coupling study, being able to do the capillary to arterial prep ex vivo, we put ourselves in a very, very good position to actually test and investigate how capillary can behave in CADASIL. And we did its first set of experiment using a thromboxane analog, U46619. That is a potent vasoconstrictor for arterial, but also for pericytes, and a very interesting one because it activates different pathways that recruit calcium in the pericytes. So for us, that was a really good indicator of whether or not capillaries, pericytes are actually impacted by CADASIL causing mutation. And here is some very interesting recordings in our prep. So I can say, like, this is a third of a human hair, about 20 micron. This is a quarter of it. This is 5 micron diameter. And right now, I really want to thank my technicians in the lab, Amanda Rosehart and Jackson Fontaine, for being able to conduct this experiment. Here you can see a microelectrode that Jackson is trying to impale on this pericyte membrane. This is how they look like in the electron microscope. So very interestingly, adding the U46619 compound to the bath really constrict the pericytes. But after 15 minutes exposure to TIM3 at a very, very low concentration, this is 8 nanomole, you can see two things. First, the resting diameter is higher, above 5 micron, it's 4.5, and the response to contraction induced by the for U46619 is actually smaller, switching from a 20-25% constriction to something around 10%. And like I said, constriction is linked to calcium and calcium, increasing calcium concentration. And to study this variation in calcium, we developed this mouse that express calcium sensor that is fluorescent in the smooth muscle cells and the pericytes because it's under a SMHC promoter. And you can see that's what happened when you do that, and this is really amazing. This is just an ex vivo prep with one pericyte being observed in basal condition. You can see all these flashes and all this wobbling and constriction going on. It's very, very live cells, lots of things going on. One of the issue with it creates this little motion when you want to precisely dissect the calcium signals. It really is very challenging. But technology is advancing, and what we've been able to is to work with actually Nikon and use an AI software that have been feeding with thousands of imaging and denoising approaches, so we can actually kind of stabilize it and get a better picture of what is going on in terms of calcium signals and study them. What we are really interested in are these spikes here that are very, very going on, and they seems to be mediated by IP3 receptors. So if you deplete the stores, they're completely gone. And we know that pericytes, they don't have renin receptors, so very likely that. And when we repeated the experiment with the U46619, you can see when it's applied, it's going to change the background in blue. Many things happen. First, the frequency of these flashes increase and the global calcium increase. And when we subject the same pericyte to TIM3, same thing for 15 minutes, you can see that both the frequency of the flashes actually decrease in response to U46619, but also the global calcium in the pericyte also decrease, which is completely consistent with the observation that the constriction is decreased, as I just showed you before. And so I'm going to stop here for this project, but we're really exciting and try to understand why TIM3 had this profound effect with calcium signals and what will be the implication. And so we are building this mechanism, this view that HbGF and EGFR pathway have different impact on depending on where you are on the cells. And we know that in arterial, it increases the Kv1.5 channel. We now know that it shut down the KR2 channel in the endothelial cells, and that can be rescued by PIP2. This is really the main message I want to give you today. It also happens to impact pericytes, and that's what we are going to investigate in the next year. So hopefully I can give you more updates on that next year or so. And so understanding the impact of extracellular dysregulation is helping us to be able to correct rapidly impairment in CADASIL. And PIP2 appears to be a very master regulator when it comes to functional hyperemia. And the overall question we want to answer in the coming years is really, does restoration of cerebral blood flow improve cognitive function and protects against vascular damage? So with that, I'd like to thank everybody that's doing the work. My mentor, Mark Nelson, we're still really actively working together. Osama and Masayo did a lot of experiment in this study with PIP2. Amanda Rosart and Jackson Fontaine-Pierre is in the lab, doing pretty much everything. Daniel, who is joining us soon. Nick Tsukias, the modeler from Florida International University. Andrew Tell, a longtime collaborator and friends in Paris. And of course, all the fundings, the NIH, the Center for Human Health and Research, of course, the cureCADASIL and the $1 million Backride grant. And with that, I'll thank you all for your attention, and we'll take any questions. Thank you.
Well, thank you very much. I, for one, thought it was an excellent presentation and tremendous slides, by the way. So we have a couple of questions. I think we have time to entertain them. Dr. Zhao asks actually a couple of questions, one of which I think you have answered. Let me bring it up here a second. Do notch 3 R169C mice show impaired cognitive function?
Very good question. We don't know. We don't know, and this is something that is also on the roadmap because at the end of the day, testing PIP2 and the ability to correct functional impairment, the ultimate readout is the cognitive assessment. The issue is the TG 129 is on the notch 3 R169C is on FVB background. These mice are almost blind. We don't have a lot of background knowledge on their cognitive behavior. You know, people who do behavioral tests mostly work with rats, not so much with mice, and even less with FVB mice. So that will be, I will try to really characterize this mouse from the beginning, and it's something that so far we are missing people to do the experiment and to help us to do this experiment. But yeah, great question.
A couple of questions from Yung Chang. Does it mean more mental exercise can increase blood flow?
Yeah, it's known. Absolutely, it's known that exercise has a positive impact on cognitive function, especially everything related to the hippocampus function, which is basically orientation, space, and memory. Yeah, absolutely. Exercise is good. It's good for your brain.
He also asked whether low blood pressure is more dangerous to CADASIL patients.
That's a good question, and I don't have the answer. I think for that we will need to turn to our clinician people and know whether, I mean, typically, from what we know from the more common form of SVD, is actually more the opposite, because hypertension is a huge factor in the development of small vessel disease. And it's pretty clear now that the state of our cardiovascular system when you are in your 40s, 50s, will strongly predict how your cognitive ability is going to evolve in your 70s and your 80s. It's more and more clear. So I know about hypertension. Yeah, definitely has a very, very high negative impact on cognitive and small vessel disease. I don't know more. I don't know about hypertension.
Here's a question from Ronan O'Brien. He says, it's very exciting and information presentation. Thank you. Are there any potential oral supplements to optimize PIP2 levels directly or indirectly?
Yeah, a good question. So full disclaimer here, as usual, I'm a PhD, not an MD, and nobody should ever take any medical advice from me. That's strongly forbidden. It can be dangerous for you. But like I said, it's remarkable that PIP2, so it's a phospholipid, and you can buy lecithin over the counter, and it's sometimes presented as a cognitive enhancement. And lecithin, which is basically white egg, contains precursor of PIP2. So it may actually work. It's also it's noticeable, and we were also thinking about eventually more downstream or upstream molecules. Yeah. So.
Okay. And as a final comment here, Dr. Zhao says, outstanding work. Congratulations.
Thank you.
And I will certainly echo that. I'm going to try to switch. I can get to my last slide. Well, I'll just say, while I'm waiting for the internet to catch up here, I'll just say thank you very much. This has been, I think, a great presentation. I want to thank all of our attendees. You will be receiving a very short survey of 3 questions. And so if you would be so kind as to fill that survey out and send it back to us. One question being, I think the most important question is, are there presentations that you would like to see in the future as we reactivate these webinars? And so let us know if you have ideas for topics and or speakers. So I think on that note, we'll call it a day. And again, thank you very much for attending.
Thank you so much.
Thank you.